Aloha, welcome to the Teaching Change series all about ʻŌhiʻa. My name is Blair, and today I'm here with Blaine, Louise, and Ava Brill, who are going to be giving us a tour of their lab and a greenhouse at the Institute of Pacific Island Forestry to teach us a little bit more about ʻŌhiʻa and what scientists are doing to manage and protect it. Ava, over to you.
Okay, aloha, my name is Ava. I work at the USDA Agricultural Research Service in Hilo, and we're actually right next to the Forest Service in Hilo. I am a biological lab technician, and I work mostly on the molecular side of the Rapid ʻŌhiʻa Death Project. My boss is Dr. Lisa Keith. She's a plant pathologist, and so she looks at diseases that affect plants, either a fungus or a bacteria. In our case, with Rapid ʻŌhiʻa Death, we're looking at a fungus, actually two fungi, that cause the disease known as Rapid ʻŌhiʻa Death. So you may have been told before that Rapid ʻŌhiʻa Death is caused by two fungi. I'm just reiterating that. So the fungi are called Ceratocystis leuco ʻŌhiʻa and Ceratocystis huli ʻŌhiʻa. And so in our lab, we're looking at detecting both of those species and looking at their characteristics, how they behave in a tree, how they move through the tree, how a tree might get infected with them. That's all a part of pathology, looking at the different characteristics and environmental factors and other types of factors that make ʻŌhiʻa susceptible to this fungal disease. My job specifically is to do molecular work in detecting rods.
Oh, we lost you for a second, Ava.
We have rod on Kauai. We had one tree detected on Maui, a few trees detected on Oahu, and then most of the trees and the first detections were on the Big Island. So if you live on the Big Island, you've probably seen some ʻŌhiʻa that have been affected by Rapid ʻŌhiʻa Death. Some of the symptoms are like a brown canopy. When you go into the wood, if you actually look at the wood, you'll see staining and stuff like that. So our job in our lab is to be a diagnostic. And what happens is land managers or homeowners from all over the Hawaiian Islands will actually go ahead and collect ʻŌhiʻa samples and send them to our lab. We do two things. We do a DNA test to test if rod is actually detected, and we do what's called a viability test to test if the fungus is still infectious. So I'm going to I'm going to flip my camera and I'm going to show you guys some of the samples that we've received. Okay, here I go. Okay, so we have an inbox. We have inter-island inbox and a Big Island inbox. Right now it's empty because I was in most of the week. But you can see an envelope right there. That envelope contains probably a wood sample. Right here, I decided to show you the types of samples we get. These are actual samples that I've received. So here is a soil sample. We will test rod in the soil. We have the capability to do that. This is a typical sample that we'll receive. This is what's called drill shavings. So in order for a manager or homeowner to take a good sample, they need to go into the wood. They need to actually take a hatchet and go beyond the bark, get into that wood tissue, look for staining, stick a drill in there, and collect drill shavings. So that's these. In addition, they also collect wood chips from the hatchet. You can see these kind of thicker wood chips. So we'll take those as well. Some agencies just send us a little piece of a branch. Oh, excuse me. We call these cookies. And they're literally just, you know, little discs of wood. And you can see where I have taken just right before this presentation, I was given, I'll show you guys, I'm sorry for the camera. I was given this piece of wood. So we receive all different types of samples. Sometimes we'll get a garbage bag full of wood. It just kind of depends who's collecting the sample and in what instance, you know, what sort of circumstances we're receiving the sample. So that is the wide array of samples that we're detecting, that we're receiving. And then I'm going to walk over to the lab side of things. So after we receive a sample, we have to be able to analyze it molecularly. And what that means is I have to pull whatever DNA is out of the sample. So from these samples, I have to then put enough material that fits into this sized tube. It's a very small tube. That is how we do our DNA extraction. And it's just a very small, it's only 2 milliliters. You can see how it compares with the drill shavings bag, you know, so I'm taking a small subsample of like the drill shavings, for example, and then I'm putting it in this tube. And then I'm gonna do DNA analysis. So let me walk over to our molecular side of the lab so you can see some lab things. Looks very labby in here. We have pipettes and some tips. These are some of the reagents we use. This is just kind of a typical molecular lab setup. These are actually already prepared tubes for something I have to do next week. So these were samples collected by a local land management agency. We just put a little bit tubes, we have a key and all that. And then our DNA extraction is a kit that takes about an hour and a half to run per 24 samples. Once the DNA is extracted, we then run a test called a qPCR. I don't know if you guys have been listening to some of the coronavirus news, but they're talking about PCR-based tests to detect COVID-19. We have a very similar test in terms of the chemistry. It's just specific for Ceratocystis ʻŌhiʻa and Ceratocystis ʻŌhiʻa. And all that's really doing the test is amplifying specific sections of DNA. And so for our test, it's amplifying ʻŌhiʻa and ʻŌhiʻa DNA if it's present in the sample. When we're ready to run the qPCR, we have this is a 384 well plate. It actually has 384 times. Then we add the DNA that we extracted, and then we run the test. We run the plate in a machine. The machine takes about an hour to run, and then we actually get computer results. And I just printed out what some of the results might look like for you guys. So this result, I don't know if you can see, but this result is, there's just a lot of weird squiggly lines, right? This is water. This is no DNA is present in the sample, okay? This means that the sample, we're not detecting anything in it. This, right? We actually have a peak coming out. This green colored peak is our molecular signal for ʻŌhiʻa DNA. So in this sample, for example, I'm not seeing any colors for ceratocystis for rod, but I am seeing ʻŌhiʻa DNA in green. And so this sample we might call not detected for rapid ʻŌhiʻa death, where we did get the ʻŌhiʻa DNA, but we're not seeing the other colors that would mean rod. This sample, I don't know if you can see, but we have, in addition to ʻŌhiʻa, blue is what we have designated huli ʻŌhiʻa. So in this sample, we have a positive detection for huli ʻŌhiʻa, for ceratocystis huli ʻŌhiʻa, and also for ʻŌhiʻa. In this sample, we have a detection of a different color, this sort of orange yellow. This is the color that designates ceratocystis luku ʻŌhiʻa. We also call it luku. And then we still have the ʻŌhiʻa signal here. So for this sample, we would call this positive for luku ʻŌhiʻa, and it came from an ʻŌhiʻa sample, so we're seeing that peak right there. Every now and then, we've run thousands and thousands of ʻŌhiʻa samples, and most of the time, it's just one species of rod or not detected. We have a handful of samples that are this. So we're getting all the colors at this point. You're getting the orange luku, the blue huli, and the green ʻŌhiʻa. We call this a co-infected sample, where actually the tree has tested positive for both luku ʻŌhiʻa and huli ʻŌhiʻa. So that's a very, very small number of samples, but we have gotten some samples that actually test positive for both species of rod. It's very rare, but it does happen, and this is what it looks like on our machine. Okay, well, in addition to that, so that's the molecular test. As I mentioned earlier, we do the molecular test to test the DNA, and then we also want to test viability. And what viability is the fungus still infectious? Can it still create disease? Land managers want to know this because let's say they go and sample a tree and it comes back positive for luku ʻŌhiʻa. If it's not viable, maybe they will leave the tree alone because the fungus is dead at that point and the fungus can't spread anymore, or a very low chance that it will keep spreading. If the sample tests like positive in the viability, meaning that we get fungal growth, then maybe the land managers are really concerned depending on the site, and they might think about other ways to manage the site. And the way we do that for ceratocystis is we do carrot baiting. So literally, we stick carrots in the wood. I'm about to move my picture again. Here we go. So, excuse me. Literally, we stick carrot in the wood, and we just kind of wait and see if anything grows out. Someone in the 1960s figured out that ceratocystis grows well on carrots over other fungi. So it's a really, really easy way to like culture out the fungus without having to use like agar. And unfortunately, I don't think I have any positive carrots at the moment. Sorry. To be positive, you would see a bunch of dark, sorry, Blair, I messed up. Oh, here we go.
That's okay. Well, I'll cut it out.
Okay. Or whatever. Okay,
yeah.
Can I jump right back in? Sorry.
Yeah.
I forgot I was supposed to find a carrot. Okay. In this case, actually, we have a positive carrot. I don't know if you can even. Then this bag versus the other bag I showed you would be positive for viability, whereas the other bag is negative. We had no fungal growth on it. So that is what we do in addition to the molecular testing. from this carrot baiting, we do some other follow-up things in the lab that aren't necessarily for land managers, but they're for us in the lab and for other collaborators. We can do what's called taking an isolate. So for all the sites around the islands, especially if it's a new detection, we want to collect a sample for our archives in the lab. So what that means is just holding onto the fungus like permanently, like a library holds books for historical significance and also for people to use. That's what we do. We do archiving of fungus. And so how we culture the fungus is we take those fungusy carrots, you take off a piece of the fungal structure and you put it onto a culture plate of agar. I'm going to show you guys an example of a nice looking plate. So this is what the fungus looks like in pure culture, it's called. And the blackish gray stuff is like the mycelium. So those are the long, it's kind of like a mat. And then it's hard to see, but the yellow dots are actually called ascospores, and they're sitting on top of the sexual structures called perithecia. And these yellow dots are very, very sticky, and that's what scientists think is probably spreading rod. Is like if a beetle comes by the tree and this sticky yellow stuff is sticking out, then it attaches like onto a beetle and it can move throughout the forest. That's one of the ways in which they think rod might move. So this is Ceratocystis leuco ʻŌhiʻa in pure culture. If you were to look at this under the microscope, you would see something like this. These are the perithecia. They're like these little balls with necks with yellow balls on top of them. And those yellow balls were the ascospores that I just showed you. So I can't show you under the microscope, but I can show you a printout. And then this is it like super zoomed in. You have this dark base and this long neck. And the spores travel up here and then they end up on top. They sort of glob on top as the ascospore and they get sent out. So this isn't a typical, this is a fungus, but it's not the fungus you're normally used to seeing. It's a different family of fungus. So these are called ascomycetes. The fungi that you guys are probably used to seeing are like mushrooms. Those are a completely different family. They're basidiomycetes. So this is just a different family of fungus, but it all works the same way with spores and stuff like that. So this is the culture. We use these cultures for many different things. We use them, like I said, for archiving. We have a collaborator that's looking at the genetic signatures of all these and learning about each culture's relationship to each other, doing like population genetics around the islands and seeing if we can get an idea of how rod has moved around the islands. The other thing we do is we do pathology testing. And what that is we actually make like a slurry, like a, okay, we want to see how rod affects seedlings in different experiments. And so we do a lot of experiments where we take ʻŌhiʻa seedlings that we grow in the greenhouse and we actually infect them with rod. To infect them with rod, we need to use things like this. And we actually make what's called inoculum, where we add water to this, we scrape it, and we make like an infectious little paper disc that we stick into the seedlings. And then we wrap them up and we wait and see what happens. And so where I'm gonna take you now is the, excuse me, where I'm gonna take you now is the growth chamber room. So after we infect the ʻŌhiʻa, then we leave them in a temperature and light controlled room the ʻŌhiʻa, depending on what we're looking for. And Blaine will talk more about this, but since the growth chamber is here, I wanted to show you guys. So for example, oh wait, whoops. So I'm in the growth chamber room now. Okay. We have, these growth chambers are literally just large, they're just big chambers. This is our rod, one of our rod ones, and this experiment has ʻŌhiʻa inside it that have been inoculated. So they've been, someone took those cultures, they scraped them, and they stuck them into these seedlings. A lot of these look healthy, but as you can see, this one, for example, is wilted and dying. I don't know if you can see that. Okay. This one is wilted and dying. This is dead, excuse me, this is dead. This is dead. So this experiment was just looking at ʻŌhiʻa and how it was susceptible to rot, and this is what we do in the lab. We have lots of different experiments, things like we'll look at different varieties of ʻŌhiʻa and see if there's any difference in how they succumb to rot, or if they do. This is all a part of the resistance project that Blaine's going to talk a lot more about. But this is how we take data. We look at, you know, we come in a few days a week and we make sure that they're watered, and we also look for just like wilting or and how long it takes for things to go kind of completely dead like this one. And then we let this experiment run many, many months. So we often run these things for 3, 4, 5, 6 months even, depending on the experiment. So that's kind of what we're doing in the lab, in addition to the molecular stuff and to the viability stuff and to the culturing stuff. And with that, I think I am done. But thank you.
Sounds good. Okay, yeah, so like Ava mentioned, one of the things we do for rod is test ʻŌhiʻa with ceratocystis to see how it responds to infection. And so I'm here right now in the greenhouse, and you can kind of see all the ʻŌhiʻa that we have growing out here. And so this is kind of how we make plants to do the experiments that Ava showed you in the growth chamber. So the plants in this greenhouse specifically are for a project looking for resistance to the diseases caused by ceratocystis, luku ʻŌhiʻa, and huli ʻŌhiʻa. So what we mean by resistance, if you don't know, is being sure that the plant won't die to the disease. And that's useful for a number of reasons. Particularly, it allows land managers, like the people that send Ava samples, to put ʻŌhiʻa back in the forest and basically regrow the forest, which is also very important for a whole number of other reasons that hopefully you folks have gone over. I don't know how much was covered before about the different species or varieties of ʻŌhiʻa, but there's a bunch of species of ʻŌhiʻa throughout the state. On our island, we have one species, and that's Metrosideros polymorpha. Beneath that, there's a whole bunch of varieties that grow in different areas of the island. We have, on record, there's 5, if I remember correctly. And what the Resistance Project is doing right now is going to different areas in East Hawaii, so in Puna and in Hilo, and going to areas that have been heavily impacted by rod, and looking for survivor trees, so trees that are still alive in these areas that have been wiped out by rod, where all the other ʻŌhiʻa have been wiped out by rod. And so we are finding survivor trees and taking cuttings. So cuttings are the tips of branches and other parts of the tree where there's leaves that we can clip off and bring it back to our greenhouses here, and we dip them in a product that contains a rooting hormone. So a rooting hormone is a chemical that when it touches the plant on the stem will cause the plant to send out roots, or that cutting to send out roots. So we can basically make a whole nother growing plant from these little pieces of branches that we can bring back. What that allows us to do then is grow them in the greenhouse. So all the plants you're seeing here are from cuttings that we took from trees out in the field. And we can bring them back, grow them up, and test them, like how Ava showed you, to see if those trees out there in nature that are surviving in these heavily impacted areas are resistant to rot, and that's why they're alive, or perhaps maybe it just didn't get infected, and so that's why it could be alive. So what I kind of want to show you guys, I'm gonna switch my screen too now, is the greenhouse. So I showed you once before, we have all different ʻŌhiʻa plants from different areas on East Hawaii, and I can kind of show you each process of how we grow things. So when we bring back cuttings from the field, they get cleaned and they get dipped in the rooting hormone, and then what happens is we grow them in these little pots here like this, and they're put into this material called vermiculite. So vermiculite is not, it's not dirt, it's not any kind of soil. I'm actually not sure where vermiculite comes from, but it's not very nutritious for the plant. It just acts as a substrate for the cuttings to grow into. So it drains very well. It looks almost like sand, and the water doesn't sit in it for very long. So that's a good thing, because as these plants are in the material and getting watered, they need some water. Those ends that have been cut from us taking it out off of the tree are open wounds that could be colonized by other diseases. And so you can kind of see we have ones that have died. I don't know if there's any others, but we have some that have just not survived the cutting process. And that happens. That's just a part of the method, but it's not due to rot. It's due to other things in the greenhouse, other pathogens, or they just didn't form roots at all and just and died. But this is how we get those cuttings started into new plants. So from here, as they get older, they may start sending out new shoots on the top and start looking like actual plants again. And after 2 months, we bring them out and we'll start to grow them out here on the bench. So obviously it's a lot sunnier out here than it is in our little chambers that have shade cloth on it. The shade cloth is to keep those cuttings cool, and with the sprinklers on, it keeps them moist, so that root growth is promoted really well in there. But once the roots start growing, the plants need sunlight to survive and to produce sugars and the things it needs to survive. So that's when we bring it out after 2 months usually, like I said before, and we put it into potting media. So this stuff is starting to look more like soil, but it isn't actually the soil you might find in your backyard or on the ground. This is a mix that we buy, and the ʻŌhiʻa that have rooted start to go in here so that they can continue to grow and get stronger. So this is what it looks like when it goes here, and you can kind of see, I don't know if you can see it very well, but we do have a little tiny root. I don't know if you can see it right there, but these things are starting to make roots and grow stronger. So from here, they kind of sit, form new shoots as well, like this, and continue to grow. And over time, they start to turn from this stuff into these bigger plants. And these, of course, look more like maybe what you would think an ʻŌhiʻa plant looks like when it's young. And so over time, we grow these plants to get even bigger. And soon you start getting plants that are in those same size pots, but are just humongous. And so the whole point of us growing these plants from different places is to grow them up and test them, right? So as they get bigger, they get thicker stems. Like this one here is very thick here, about the width of a pencil. And that's perfect for testing because, as Ava mentioned in the growth chamber, we're putting Stratocystis in the plants. So we'll make a little wound on the stem here where we cut it with a little knife. That makes a wound that we can then put the inoculated paper disc into the wound, and we close it up and seal it up with some stretchy film, and that allows the fungus to colonize the plant and sometimes causes death, like how Ava showed you. And so, as I mentioned also before, there are different types of ʻŌhiʻa, so I kind of wanted to show you folks the different types of ʻŌhiʻa we have and how maybe you might be able to distinguish them. I don't know if you folks went over that yet. So one type that we have is called ʻŌhiʻa, and so these are all the same species of ʻŌhiʻa. So they are able to make seeds with each other, but they just look a little different, and they are from different places on the island, and so they're called different things. So this is Metrosideros polymorpha ʻŌhiʻa, and so these leaves, I don't know if you can see, are pointy, kind of pointy on the ends. They're pretty small, and the leaves kind of stick really close to the stem here. And I don't know if you can really tell either, but they are hairy. They got this gray fuzz on the bottom, and when I rub it, it doesn't come off. So that's a sign that this is a true ʻŌhiʻa ʻŌhiʻa, that the hair sticks on like that, and this leaf shape as well. So this is from the lower elevations of the Big Island. You find ʻŌhiʻa, and it likes to grow where it's hot, where there isn't a lot of rain, and a lot of times you find them on new lava flows down low elevation. Another type that we have, if I can find it, is Glaberrima, and this is another type of ʻŌhiʻa, and I don't know if you can tell, the leaves don't look as pointy, and the leaves look a little darker green. And so these leaves are a little darker green because they don't have hair. And so that's why this ʻŌhiʻa is called Glaberrima. In the botany world, where there's all sorts of terms for all sorts of things that you and I probably have never thought about, the term for not having hair is glabrous. And so that's why this one is called Glaberrima, because there's no hair on the bottoms or on the tops of this leaf. There are some little bugs here, but no hair. So like I mentioned before, sometimes the ʻŌhiʻa, even though they're different, will breed with each other and create seeds. And so we get what are called hybrids. So a hybrid, I believe these are hybrids right here. They look kind of intermediate, where they aren't super pointy, but they have that point like in ʻŌhiʻa. They're not super hairy either, but they do have a little bit of hair. And the trick to seeing if it is or isn't a hybrid is, I don't know if you can see this hair here, it's kind of gray, but if I rub it, the hair comes off. Can you kind of see how that hair kind of rubs off like that? That is how you know, is a pretty good way to tell if your plant is a hybrid between Glaberrima and ʻŌhiʻa, or Glaberrima and another variety of ʻŌhiʻa called polymorpha. And that variety, polymorpha, grows up higher on the mountain. It's a high elevation variety that likes the cold. So these three different varieties all grow in different places, and they all serve different ecological functions. But that is something we really want to test, are all these different types of ʻŌhiʻa, see if where they've grown, and you know, all the history behind these plants growing where they've grown have led to adaptations, and we want to see if any of those adaptations make it better at surviving rod, and what those adaptations may be, and how we can use them for other varieties that maybe don't have those adaptations, and maybe try to breed those plants to get that resistance in there. So the cuttings project is one project we do, and I'll just show you really quickly one more thing that we focus on too is not just producing cuttings from ʻŌhiʻa, but we also will produce seedlings as well. And so in here, this is the same substrate that we grow the bigger, more fully developed cuttings in. And so it's kind of like dirt. But what we do is when we go and collect seeds from the field, they're really tiny, almost like dust or sand. They're very small. And ʻŌhiʻa seeds, because they're so small, are dispersed by blowing in the wind. And so if you can kind of think maybe about how ʻŌhiʻa might have gotten to Hawaii, which is one of the most isolated island chains in the world, it probably blew from an island that was close to us on the wind and came and got here and grew and turned into all the many species and varieties that we have. So we collect those seeds and we put them on this potting mix, and they start to turn into these little tiny guys right here. So they're small. They're really small, and it takes them a long time to kind of get bigger to the size I showed you before that we would inoculate with. But the good thing about this is that we can create many, many plants without very much effort, unlike the cuttings where we have to basically cut all the plants that we want to make and hope that they root. So these plants will also get raised until they're that size, and we'll test them. These are also seedlings from some of the trees that we took cuttings from too. So it's kind of a two-way approach to the situation. So with these material types, we're hoping that we can, let me switch back. Where is it? Oh. So with those two material types, what we're hoping to do is screen them, test them with the fungus, and see which ones survive. And those survivors then, we can't be 100% sure that they're resistant. We usually will test many, many times to get that, as I get blasted by the sprinkler. We usually have to test ʻŌhiʻa a few times, not only in a greenhouse like this or a growth chamber like Ava showed, but also out in the field too, where the plants maybe might go. And that's just to really be sure that what we're seeing is not just something that happened randomly and that's why it survived, or maybe you messed up the inoculation. You test it multiple times to be sure that the plant will always resist infection so that it can live happily out in the forest and we don't have to worry about it dying from rot. And so I think that's it for the greenhouse.
Thank you, Blaine. Thank you,
Yeah.
Ava. I have a question for you both. Do you guys have any results from your experiment to find trees that are resistant to rod, or are you still in the preliminary stages?
So, I mean, a lot of it is preliminary, and I'm going to go outside so it might get really bright really fast. But we do have some ʻŌhiʻa that have survived experiments, and so we like to obviously not cut them down and check them out on the inside. We like to save them because you never know if they're actually resistant or not. So what we do is we save those plants and we raise them to try and test them again. So I don't know if you can see this sign here. It says these trees have been inoculated with Stratocystis. So these plants in the back of the greenhouse here are growing to be propagated again, whether that's from cuttings, like how I showed you in the greenhouse, or if they end up flowering and we can produce seed, we might test that. But the goal is to get to the stage where we can test these plants that have survived infection again. So these plants, most of them have survived being infected, I believe over 2 years, probably a little bit more. So the fungus has been in them for 2 years at least, and you can see that they don't look dead like how Ava showed. They look nice. They maybe aren't super healthy, but we have them out here to hopefully get them more healthy and grow them up and test them again. Oh, look, one died. That could have been from the process of potting it up, because that's also really stressful for plants as well.
The transfer.
Yeah, it's really stressful, especially if it's coming straight back out into full sun, unlike the nice shadiness of the greenhouse. It can be harsh, but that's nature, you know. It's the real conditions of the field. So these plants have been inoculated. They've survived for several years now, which is longer than we would expect most plants to survive. And we hope to learn more about them and see where that takes us. But all of our other projects that we have and the formal resistance project are still really preliminary, and we don't have any super definitive results as of yet.
So would your goal then to be, once you've really established that, you know, you have some plants that are resistant to rod, to grow a certain amount to outplant them on Hawaii Island or just East Hawaii Island or statewide?
Yeah, so I don't know if it's better for me to change back to this way, but better lighting. So the goal would be to, so first we have to test in the greenhouse or the lab, like we're talking about. And once we feel pretty confident that they show resistance in the greenhouse or the lab, what we then do is take them out to the field and see if that, you know, survivorship or that resistance persists out in the field. And if that is the case, then we can be pretty confident that that they are resistant to disease. So what we would do then is develop orchards of resistant ʻŌhiʻa. And so that is mainly so that, I don't know if you know what an orchard is, or like how we have apple orchards, for instance, where they produce apples. We'd have an orchard of ʻŌhiʻa to produce disease-resistant seed. So we can use that seed, we can give it to land managers that want to grow their own seed and plant it out wherever they want. We can also do experiments with breeding the different resistant trees to see how resistance is inherited by future generations of ʻŌhiʻa. And the other thing too, thinking about disease-resistant orchards, is that, like I was telling you about the different varieties of ʻŌhiʻa that we've tested or that we have here, they end up, or they grow in different conditions. There are ones that grow in really hot, low elevation conditions, moist, mid elevation, and cold, high elevation. And so obviously we can't grow all of the ʻŌhiʻa in one seed orchard. We have to grow them in various places and make sure that the resistance does hold up in all different conditions, and make sure the plants are suitable for the places that people want to grow them in. Otherwise, you're just putting a plant in the ground for it to suffer.
I have learned so much. I was wondering if Ava and Blaine, if you could both share a little bit about your career background. What did you do during high school and after high school to get to where you are today?
You want to go first, Ava? I'm walking back into the greenhouse. It's hot.
Sure. I know it's really hot today. Sure. So in high school, I was always interested in nature in high school, but I actually went to a liberal arts college for my undergrad, and I actually did the science program at my liberal arts college, which was like weird within the weird college. And I'm originally from the East Coast, so I did work in salt marshes, and I actually got a scholarship to go to Alaska to do an environmental internship my last year of college, and that I think is what really got me into conservation. And then I moved to Oregon for about 6 or 7 years, and I worked as a nonprofit high school teacher at a nonprofit high school. I worked as a science teacher, and then I decided to go back to school, and I wanted to do conservation biology, so I applied to conservation biology programs, and I got into ʻŌhiʻa, and I did the TCBES program at ʻŌhiʻa, and I graduated in 2015, and since then I've actually been working at USDA. I originally worked in a different lab working on coffee and macadamia nut pests, but then switched over to Lisa Keith's lab, where I can work on ʻŌhiʻa. So that's been really great. So that's my journey.
Awesome. Thank you.
Yeah, so mine's is definitely not similar to Ava's pathway. So I was raised in Kona, on this island. I went to Kona High School, and I honestly, I guess, didn't think I was going to be doing disease resistance work when I was in high school. I was thinking, I guess I was going to be a pharmacist, and so, which is obviously it's sort of a big difference. But if you think about it, I guess pharmacists deal with human diseases, and now I'm working with plant diseases, so I guess that's maybe not too far off. So I went to UH to get into pharmacy, and a year or two in, I decided pharmacy wasn't for me, and so I switched because I was already doing biology, my degree in biology. I just switched over to ecology because I like being outside as well, and you know, being in the forest or being wherever outdoors. And so over time, I ended up getting into the PIPES program, and the PIPES program is really great. Shout out to the PIPES program. They provide undergraduate students with internships at different institutions or different agencies here in Hawaii. And so I was fortunate enough to get an internship with the Forest Service. So I actually started with a research ecologist. His name is Flint Hughes, and he does a lot of ecology work. So I helped him monitor plots for ʻŌhiʻa rust. So you can kind of see how we're getting closer a little bit. We're looking at ʻŌhiʻa rust, which is a different disease of ʻŌhiʻa. It affects the leaves, and it looks completely different from rod. And I can actually show you an instance of rust. Rust looks like this. It's kind of like these little blotches on the leaves. I can't zoom in that well. My camera won't let me zoom in more than that. But in those little red blotches, there's little tiny yellowish, they're called pustules. Those are fungal spores. And just like ʻŌhiʻa seeds, they're so tiny that when water splashes them or wind blows, they move and they infect other ʻŌhiʻa trees. So they have an easy time getting into our greenhouse that's just green on the sides. And how wet and cool and moist it is in here, it grows really well. So I worked on that for a little bit with Flint. At the same time, Flint and some other researchers like Ava's boss, Lisa Keith, who is still kind of my boss, and JB Friday all started looking into rapid ʻŌhiʻa death, which was just starting. And I think this was in 2014 where things started to be noticed. And so meanwhile, somebody who was also under Flint was working on rod, so we'd help each other out with our projects. So I'd help, his name was Kaisen, I'd help Kaisen look at his work on rod at Lisa's lab, and he'd help me with the rust plots. And eventually, after PIPES was done, I just reached out to Lisa and asked if there's any opportunities to continue on, and she was able to bring me on, and I learned all my pathology work through Lisa, so I didn't go to school to become a pathologist. And then all of that ended up with me going back to UH to do a master's degree at TCBES. So Ava and I are products of TCBES. And I was able to get my master's studying whether or not different varieties of ʻŌhiʻa have resistance to Ceratocystis leuco-ʻŌhiʻa infection. So this whole resistance project that I've shown you it stems out of my work from my master's project, but has become a much bigger deal with many more people involved. So it is great to see where this is all going. So if I can give you folks one bit of advice, it would be to not maybe expect to go where you might think you're going. Life has weird ways of taking you off course, but you end up sometimes in a better place because of it. So enjoy the ride, go along with the ride, and try your best. Don't just let things go where they're going to go without any effort. Put in all the effort you can and see where it takes you.
That is excellent advice. I second that. Well, thank you both so much, Ava and Blaine. That was excellent and so much amazing information about ʻŌhiʻa and the work you're doing to mitigate rapid ʻŌhiʻa death. I certainly learned a lot, and I really appreciate you sharing your professional development pathways too. I think that's great to start talking about while you're in middle school and high school. So if that's it from you guys, that is all the questions I have. Is there anything else you would like to say?
No, thank you.
Thank you both so
Yes, thank
much.
you.
Bye.
Okay, aloha.
Take
Aloha.
care. Bye-bye.